Respiration means we have 3 steps in respiration, right now. First one is external, second internal, third one is cellular respiration. External and internal, these two processes we will read in human respiration, human physiology. And this cellular respiration we are going to read this in this chapter. Cellular means we have two processes. Which one? Glycolysis and Krebs cycle, right? So glycolysis, where does glycolysis occurs? In cytoplasm. And Krebs cycle in mitochondria. Krebs cycle means oxidation, right? That is aerobic respiration. All of us breathe to live, okay? But why is breathing so essential? Breathing is common sense. We said them. Breathing is the respiration. Inhale and exhale your gases. Essential. What happens when we breathe? First, we have 3 questions. First, why is breathing so essential in life? Why it is important? Oxygen. Why oxygen is important? To formation of ATP. That way to produce energy, right? Breathing is essential for producing energy in the form of ATP, right? And what happens when we breathe? What happens when we breathe? Exchange the gases. CO2 is released and oxygen is absorbed into the blood. Also, do all livings, including plant and microbes, breathe? Yes, all, because all needs energy. If so, how? Now the question, how they breathe? So different organs they have different type of respiration. Some animals have cutaneous respiration like earthworm. Cutaneous means skin through. Some have tracheal respiration like insects. Scorpions have buccal lungs, right? Some have gills, aquatic. We have lungs. In plant, direct diffusion is there, right? Stomata, opening of stomata, that means exchange of gases. Carbon dioxide released into the atmosphere. But in case of plant, they use carbon dioxide in the process of photosynthesis. So they did not release CO2 because they use CO2 in the process of photosynthesis. We release CO2. So they that means absorb oxygen through stomata, right? Lenticels are also, that means in bark, lenticels are there. The cells which are present in barks, they absorb oxygen through diffusion. Which structure is there? Lenticels are there. 2% of exchange of gases is through lenticels. 98% through stomata. Now here, all living organisms need energy for carrying out daily life activities. They need energy, be it absorption, transportation, movement, reproduction, or even breathing. Breathing also need energy, because without energy, no activity is there. Right? For work, we need energy. All these are processes: absorption, food absorption, transportation, movement, reproduction, and even breathing. Where does all this energy come from? This energy comes from respiration. They come from respiration. We know we eat food, and food, that means we have which food? We have 3 components in foods. Which one? Carbohydrate, lipid, and fat. These are 3 components, right? And for vitamins, vitamin is not used in the formation of ATP, right? The vitamin is not digested. Minerals are not digested. Water is not digested. Fibers are not digested. Only 3 food steps are digested. That is carbohydrate, fat, and protein. And these 3 components we use for energy, right? The first component, who is the preferred? Carbohydrate is preferred, right? Here, we know we eat food for energy. But how is this energy taken from food? That is the question. This energy is taken from food through respiration. How is this energy utilized? How it is utilized? It is utilized in the form of ATP, right? ATP is converted into ADP and energy is released. When energy released, this energy is consumed by the process. Do all food give the same amount of energy? No. Not give. Okay. Do plant eat? They did not eat. They need not to eat food because they prepare their food from sunlight, right? Where do plant get their energy from? They get the energy from sunlight. Sunlight, na? They trap this photon, sunlight energy, into the food, right? And microorganisms, for their energy requirement, do they eat food? They need not to eat food. If there are some parasites, so they directly absorb glucose, other things, and metabolic activity, they produce ATP. If they are like cyanobacteria is there, so they prepare their food in the sunlight. You may wonder at the several questions raised above, they may seem to very disconnected. But in reality, the process of breathing is very much connected to the process of release of energy from food. Let us try and understand how this happens. That means what here? Okay, some eat food, plant did not eat food. So they have different type of breathing process. But one thing is common. In cellular respiration, everything is. Glycolysis is common in all the organisms. And glycolysis is universal. Universal because it is either aerobic or anaerobic. Animals or organisms, they have glycolysis. After glycolysis, some like yeast, fermentation is there. Fermentation is there. In fermentation, yeast forms ethyl alcohol plus CO2. Our muscle cells also, they form lactic acid. Right? In our normal cells, this pyruvic acid goes into the Krebs cycle, mitochondria. But glycolysis is same. That means the molecular basis of respiration is same in all the organisms. All the energy required for life process is obtained by oxidation of some macromolecules. Macromolecules means carbohydrate, protein or fat, right? That we call food. Only green plants and cyanobacteria can prepare their own food by the process of photosynthesis. I already told you. Cyanobacteria, autotrophic blue-green algae. They trap light energy and convert into chemical energy. In photosynthesis, we read this one. That is stored in the bonds of carbohydrate, glucose, sucrose, starch. Right? Glucose, this one is monomer. This sucrose, it is disaccharide. Right now? Disaccharide. Sucrose means was glucose plus fructose. Right? Starch is a polysaccharide. Right? Starch contains two types of, that is one is amylose, another one is amylopectin. Amylose plus amylopectin, they form starch. These are two forms of starch. Amylose means what? Amylose means they have coiled structure unbranched. And amylopectin means coiled structure but branched. Glycogen is also branched but highly branched. Glycogen is highly branched, right? In potato, starch is there. In potato, 80% is amylopectin and 20% is amylose, right? So these are glucose, sucrose, starch. Now you understand. Monosaccharide, disaccharide, polysaccharides. We must remember that in green plant too, not all cells, tissue and organs photosynthesis. All cells or all organs did not. That means this occurs the photosynthesis process. Only green part of the plant. Only cells containing chloroplast, that means green cells, that are most often located in the superficial top layer, right, carry out photosynthesis. Hence, even in green plant, all other organs, tissues or cells that are not green, they are also living, they need energy, need food for oxidation, and this food comes from this green part of the plant. Hence, food has to be translocated, that means transported to all non-green part. Transportation is essential here. Animals are heterotrophic. We are dependent upon the plants for food. They obtain food from plants directly, they are called herbivorous, or indirectly as for carnivorous. Saprophytes means organisms which absorb their food through decaying matter, right? Like fungi are dependent on dead and decaying matter. What is important to recognize is that ultimately all the food that is required for life process comes from photosynthesis. We know very well this. So the plants are called producers, right? So this chapter deals with cellular respiration. It is given here. Cellular respiration deals with now this mechanism of breakdown of food materials within the cell to release energy. What is cellular respiration? That is mechanism of breakdown of food materials within the cell to release energy and the trapping of this energy for synthesis of ATP. Similar, that means simply you can say breakdown of food materials and the formation of ATP. This is called cell respiration. Photosynthesis, of course, take place within the chloroplast. We know very well chloroplast. In the eukaryotes, because they have chloroplast, but in prokaryote, no chloroplast is there. Whereas the breakdown of complex molecules to yield energy take place in the cytoplasm and in the mitochondria. Chloroplast is responsible for photosynthesis, and mitochondria and cytoplasm responsible for breakdown of this food which is formed by the chloroplast. Also only in eukaryote, because mitochondria is present only in eukaryotes, right? The breaking down of C and C, CC bond, carbon-carbon bond, of complex compound through oxidation. Now look here, if it is a glucose, we have glycosidic bond. If it is a fat, we have ester bond. If it is a protein, we have peptide bond, and they are all CC bonds. So here it is given only C, carbon-carbon bond, right? To complex compound through oxidation within the cell, leading to release the considerable amount of energy, and that is called respiration. Now here, what is respiration? It is given. What is respiration? Breakdown of carbon bonds, right, of the complex compound. Complex compound means either carbohydrate, protein or fat through oxidation. Oxidation is also called as burning, right? Oxidation by using of oxygen within the cell, leading the release of considerable amount of energy, that is called respiration. Correct? The compounds that are oxidized, and they are called as respiratory substrates. Usually carbohydrates are oxidized to release energy. First carbohydrate oxidation, but protein, fat, when the carbohydrate is not present or absent, then fat is used, and if fat is absent, then protein is used, right? But protein, fat and even organic acid can be used as respiratory substances in some plants under certain conditions. They have some conditions. During oxidation within a cell, all the energy contained in respiratory substrate is not released free into the cell or in a single step, but it is released in series of slow stepwise condition, right? Burning and respiration both are same, but in burning all the energy released, but in case of respiration energy is released in step by step, right? It is released in the series of slow stepwise reaction controlled by enzymes, and it is trapped as chemical energy in the form of ATP, adenosine triphosphate. Hence, it is important to understand that the energy released by oxidation in respiration is not used directly, but is used to synthesis ATP. That means breakdown of glucose. Why? What is the aim? What is the target of breakdown of glucose? To formation of ATP, not to release energy, right? Or rather cannot be, is given na. Sometimes it is used, directly it is used, but after the formation of ATP. Like in glycolysis, we have 4 ATP are formed, but we gain only 2 ATP because 2 ATP is immediately used, right? But this energy is used after the formation of ATP. Without formation of ATP, our cells cannot use energy. Hence, ATP acts as energy currency of the cell. Sometimes it is called energy coin of the cell. Coin is also currency. This energy trapped in ATP is utilized in various energy requiring process of the organisms, and the carbon skeleton produced during respiration is used as precursors of biosynthesis of other molecules in the cell, like alpha ketoglutarate acid form. Alpha ketoglutarate acid, it has many functions. It is the intermediate substrate of Krebs cycle also. It is also a fatty acids which is used in the formation of other large fatty acids like cholesterols, formation of cholesterols or vitamins. Alpha ketoglutarate, it is also used, right? So alpha ketoglutarate, it becomes precursor of some other molecules, right? So do plant breathe? Well, the answer of this question is not quite so direct because they have no specific breathing organs, but they also breathe. Plant require oxygen for respiration to occur, and they also gives out CO2. Same thing is there. This plant require oxygen for respiration to occur, and they also give out carbon dioxide. They also release carbon dioxide, but this carbon dioxide, which is released by the cell, it is trapped by which one? Calvin cycle, right? And this one is used in Calvin cycle. In night, sometimes it is said, don't sleep under the tree. Why? Because in night they release CO2. Because in night no photosynthesis is there. So the plant, when they respire, the CO2 is released, and this is released into the atmosphere also, right? But in day, this energy, this CO2, it is used by the plant. Hence, plant have system in place that ensure the availability of oxygen. Plant, unlike animals, have no specialized organ for gases exchange like lungs, but they have stomata, 98% stomata, and lenticels in bark stem for this purpose. They have 2% from lenticels. There are several reasons why plant can get along without respiratory organs. The first one thing is these statements are important, because in this statement, why plant can get along without respiratory organs? Why no respiratory organs is developed in evolution? Why not? Because they need not to. Look here. First, each plant part take care of its own gas exchange needs. Every cell is responsible. That is stomata, the whole leaf. Lenticels for bark sound, not for whole body. And our lungs, it is responsible for whole body oxygen. There is a very little transported of gases. They have no transportation of gases. Little transportation of gases, which is in the soluble form of water or other things. But in our case, we have transport system. We have hemoglobin, right? Or there is a very little transport of gases from one plant part to another. Second, plant do not present great demand for gas exchange, because we need more energy. We have movement, other things are there. But plant, they have no movement, no other things. They need not more energy. Root, stem and leaves respire at rate far lower than animals do. Only during photosynthesis are large volumes of gases exchanged, and each leaf is well adapted to take care of its own needs during this period. So when photosynthesis, they need more CO2. So this photosynthesis for CO2, they have stomata. When cells of photosynthesis, availability of O2 is not a problem in these cells, since O2 is released within the cell in the process of photosynthesis. So plant have photosynthesis, O2 is already released in photosynthesis. So they use this O2 in respiration. Excess CO2 is also there. So the excess CO2 they release in the environment. Excess O2 they release into the environment. So they have already oxygen which is released in photosynthesis, and they use this oxygen. Third, so we have to, what are the two things given here? First reason, why the plant have no organs? First, what is given? Little transportation of gases, right? Second, they need less amount of oxygen, and oxygen is also released within the cell, right? And third one is the distance that gases must diffuse even in large bulky plant is not great, because each cell is directly exposed with the air. Look at distance that gas must diffuse even in large bulky plant do not great. Each living cell in a plant is located quite close to the surface of the plant, surface part of the plant. Very thick stem is there. What is in the middle part, the center part? Which type of cells are there? They are dead cells. Wood is there. Wood means xylem, right? Now, so xylem is there. So xylem is made up of dead cells. They need not to energy. Outer bark is there. The biter is directly in contact with the air. It is given na. Each living cell in a plant is located quite close to the surface of the plant. This is true for leaves also. You may ask, but what about the thick woody stems and roots? In stem, the living cells are organized in a thin layer inside and beneath the bark. Beneath the bark, living cell is there. But then after we have wood, and wood is non, right? They also have opening called lenticels. So these cells get oxygen from lenticels. The cells in the interior are dead and provide only mechanical support. This wood is made up of dead cells. They need not to oxygen. Thus, most cells of the plant have at least part of their surface in contact with air. So they need not any transportation or any organ. This is also facilitated by the loose packaging of parenchyma. Parenchyma, they have intercellular space is there. Intercellular space, they have air. Stems, leaf, stem and roots, which provide the interconnected network of air spaces, right? So we have three things here given why they need not organs. First, little transportation. Second, they need less amount of oxygen. And third one is given, they are directly contact with the air mostly. The complete combustion of glucose which produce CO2 and H2O as end product yield energy, most of which is given out as heat. Okay, it is given as C6H12O6, that is glucose, 6O2. Now it is 6 CO2 is released, H2O plus energy is released, right? So this is the complete oxidation of glucose, right? And this one is aerobic respiration because oxygen is there. If this energy is to be useful in the cell, it should be able to utilize to synthesize other molecules that the cell requires. The strategy that the plant cell used is to catabolize the glucose molecule. What is catabolize? We have two processes: catabolic activity and anabolic activity and catabolic activity, right? And both are said, we commonly said metabolic activities. So catabolic activities means breakdown of large molecules. Anabolic means formation of large molecules, right? Which one is energy yielding, and which one is which produce energy and which consume energy. Breakdown means bond are break. When bond breaks, energy is released. So catabolic activity, energy is released. Anabolic activity, energy is consumed, which is trapped into the. That means what? Photosynthesis. Catabolic or anabolic? Normally, generally, what we say about photosynthesis, it is catabolic or anabolic? Anabolic, because CO2 plus H2O, it makes a molecule, right? And this photosynthesis, normally it is said anabolic, but it is amphibolic. Why amphibolic? Because ATP is also formed. ATP is also formed, so it is amphibolic. Now here, the key is to oxidize glucose not in one step but in several small steps, enabling some steps to be just large enough such that energy released can be coupled to ATP synthesis. Okay, here the story of respiration is given here. During the process of respiration, oxygen is utilized. First thing is here, oxygen is utilized, respiration, and oxygen is utilized, that means it is aerobic respiration. We are talking about aerobic respiration, right? And carbon dioxide, water and energy are released as a product. What are the product? That is carbon dioxide, CO2, H2O and energy. The combustion reactions require oxygen, but some cell lives where oxygen may or may not be available. Can you think of such situation and organisms where O2 is not available? An example is there. Anaerobic bacteria are there, right? In case of yeast also, fermentation. Muscles, when we have heavy exercise, then our muscles cannot get sufficient amount of oxygen. So they also starts formation of lactic acid. It is also in the absence of oxygen. So there are sufficient reasons to believe that the first cells on this planet live in an atmosphere where oxygen is absent. At first, oxygen is not there, right? No? So first organism which is present in our, that is which evolved in our Earth, This one is heterochemotrop. It is not autochemotrop, heterochemotrop. It cannot prepare its foods in sunlight. It only prepare food, it only obtain food from chemicals. After heterochemotrops, this chemoautotrop comes. And chemoautotrop, then autotrop, cyanobacteria comes. Right? Lacked oxygen. Even among present day living organisms, we know the several that are adapted to anaerobic condition. Anabacteria are there. Some of these organisms are facultative anaerobes, while in other, the requirement of anaerobic condition is obligate. We have two types of bacteria, either facultative aerobic or obligate aerobic. Right? What do you mean by facultative or obligate? Yes. Obligation means what? If oxygen is absent, they can survive in the absence, anaerobic condition. Right? Facultative means they cannot survive in anaerobic condition. That is one. Right? In any case, all living organisms retain the enzymatic machinery to partially oxidize glucose without the help of oxygen. Now here, in case of anaerobic respiration, glucose is partially digested or partially breakdown. Right? This breakdown of glucose to pyruvic acid, now it is given, it is called glycolysis, and it happens in cytoplasm. Now glycolysis is given. How many steps are glycolysis there? We have 10 steps, right? In which maximum are reversible, but 3 are irreversible, right? So 3 means what? First, 1, 3 and 10. First step, third step and 10th step. They are irreversible, right? The term glycolysis has originated from the Greek word glycose. Glycose means what? Sugar, right? And lysis means splitting. That means sugar is splitting. The scheme of glycolysis was given by Gustav Embeden, Otto Meyerhoff and J. Parnas. Sometimes we said them, what say we said them? EMP pathway, right? And shortly we said them Embeden, Meyerhoff, Parnas pathway, right? And is often referred to as EMP pathway. In anaerobic organisms, it is the only process in respiration. Anaerobic respiration, this one is the only process. Glycolysis occurs in the cytoplasm in the cell and is present in all living organisms. That means it is universal, right? We can say it is universal. In this process, glucose undergo partial oxidation to form 2 molecules of pyruvic acids. So one glucose break down partially and forms 2 pyruvic acid. Glucose is hexose sugar, 6 carbon molecule, and pyruvic acid 3 carbon molecule, right? In plant, this glucose is derived from sucrose. Plant sucrose. This one is a disaccharide. Sucrose is also called as transported sugar, cane sugar. We can see this one, right? Why it is called transport sugar? Because for transportation, plant use only this sucrose. It has starch. First, it break the starch into sucrose, then it transported. Because sucrose is soluble in water, it can easily transport it. Starch is not transported, right? The plant, they use sucrose, and sucrose means first they have to break down the sucrose into glucose and fructose, right? So for the breaking down of glucose into glucose and fructose, right? Which enzyme is used? Invertase, right? The glucose is derived from sucrose, which is end product of photosynthesis, right? So here it is also given. What is the end product of photosynthesis? Sucrose is the end product of photosynthesis, right? Not glucose is the end product. Sucrose is the end product of photosynthesis, right? Or from storage carbohydrate. Storage carbohydrate means plant is starch. Sucrose is converted into glucose and fructose by the enzyme invertase. It is given also by the enzyme invertase is there. And these 2 monosaccharides, which one? So glucose and fructose. What is the difference between these two isomers? They are isomers, right? So they are example of anomers. Read in biomolecules, anomers and apimers, right? First carbon change of that is when the place is changed, H and OH, right? In first carbon, the first carbon is difference is there, that is called anomers. So they are the example of anomers, right? Second thing, glucose is dextrose, dextrorotatory sugar, right? And dextrorotatory means what? Right-handed rotation is there. And fructose, it is also called as levulose because it is anti-clockwise rotation. It is called levorotatory sugar, right? Both are isomer by the enzyme invertase, and those two monosaccharides readily enter the glycolytic pathway. What is glycolytic means what? Glyco means glucose, lytic means lysis, splitting pathway. Glucose and fructose are phosphorylated. Phosphorylated means phosphorus is attached. Phosphoric acid is attached. Phosphorylated to give rise to glucose 6-phosphate. First step, glucose 6-phosphate is formed. This step is irreversible. Glucose 6-phosphate cannot convert into glucose. That is by the activity of enzyme. Which enzyme is there? Hexokinase. Because this one is hexose sugar, right? So hexokinase. This phosphorylated form of glucose then isomerizes to produce fructose 6-phosphate, right? Now fructose 6-phosphate is then converted into fructose 1,6-bisphosphate. So 6 means what? Sixth carbon of, look here, where is 6? This one, 6. So sixth carbon of glucose is bind with H3PO, that is phosphoric acid, right? And 1,3 means what? First carbon of glucose and sixth carbon glucose. Both are binds with phosphoric acid, H3PO, right? Bisphosphate by the activity of the enzyme hexokinase. No, where it is? Subsequent steps of metabolism, various steps of glycolysis depending. Okay, what in glycolysis a chain of 10 reactions, it is not given here, right? But in chain, this chain it is given. Look, glucose 6 carbon molecule. Now this phosphoric acid comes from ATP. Now 1 ATP is used. Which enzyme is used here? Hexokinase enzyme is used. Now glucose 6-phosphate, which is 6 carbon molecule, is converted into fructose 6-phosphate, right? Which enzyme is there? Now it is isomerase enzyme, because isomer is there. Isomerase. Now fructose 6-phosphate is converted into fructose 1,6-bisphosphate. Another ATP is used. Now 2 ATP are used, and this one is the third reaction. This one is also irreversible, right? Now fructose 1,6-bisphosphate is formed. This fructose 1,6-bisphosphate, it is converted into triose phosphate. Triose means 3 carbon molecule. We have 2, PGAL and DHAP, phosphoglyceraldehyde and dihydroxyacetone phosphate, right? Now 3 glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This one is ketone form. This one is aldehyde form of sugar, right? Which enzyme is there? Aldehyde is formed, aldolase enzyme, right? Now here one very important thing, NADH2 is formed. This 2H comes from this glyceraldehyde. Dihydroxyacetone is not converted into, first dihydroxyacetone converted into glyceraldehyde, then it comes into, right? NADH is formed. How many NAD is formed? 1 NAD from 1 glyceraldehyde. Now we have 2 glyceraldehyde, so 2 NADH is formed, right? In which step it is formed? Third, fourth, fifth. Which step? Fourth or fifth? Which? Fourth step. 1, 2, 3, 4, fifth step, right? So fifth step, NADH is formed. On 2 NADH is formed. Now this, after then what happens here? 2 multiply, but means 2 molecules of triose bisphosphate. Phosphate means what? It is BPGA, short form we get them, right? Now 1, 3, because it is a triose sugar, we have 3 carbon molecules on it. For last carbon is 3. First carbon is first. 1, 3 by phosphoglyceric acid, BPGA, right? So which phosphoglyceric acid is formed? Which is 3 carbon molecule. Now look here. How many phosphate are there? 2 phosphates. One first carbon and third carbon. Now one ATP is formed here. So one phosphoric acid from this glyceric acid come and bind with ADP and forms ATP. How many ATP is formed in this place, in that place? 2 ATP through this 2-1, right now? Now triose phosphate, 3 phosphoglyceric acid. So first who release? First carbon release the phosphoric acid, right? First come to the last carbon. Look, one ATP come to the 6th carbon. Then another ATP come to the first carbon. And when going, releasing, so first who release? First carbon release. And then, right? So first come, last go. Rule is there. So here 3 phosphoglyceric acid. Third carbon has phosphoric acid. Now what happens here? Mutase enzyme, right? In the presence of mutase enzyme, what happens? This third carbon, the phosphoric acid which is on third carbon, it comes to the second carbon and it forms 2 phosphoglyceric acid, 2PGA, right? And then enolase, in the presence of enolase enzyme, what happens here? Phosphoenol pyruvate is formed, PEP is formed, right? And one H2O is released. Here one H2O is released. And in last step, this one is last. Pyruvic acid is formed and one ATP from one PEP, that means 2 ATP from 2 PEP is formed. How many ATP is formed? 2 from here, 2 from here, 4 ATP is formed and 2 NADH is formed, right? So this NADH, where it go? It go in electron transport system for the formation of 3 ATP. 1 NADP forms 3 ATP, 1 FAD forms 2 ATP and 1 GTP forms 1 ATP. 1 ATP means 1 ATP is there, right? So here 4 ATP are obtained, in which 2 ATP is already used. Now net gain is only 2 ATP, right? Steps of glycolysis given here. The fructose 1,6-bis phosphate is split into dihydroxyacetone phosphate and PGAL phosphoglyceraldehyde. We find that there is one step where NADH2, right? H plus H, we write this one, is formed from NAD, right? This is when 3-phospho is given here. How this NADH is formed? This is when 3-phosphoglyceric acid is converted to 1,3-bis phosphoglycerate, BPGA, right? 2 redox equivalents are removed in the form of 2 hydrogen atoms, and these 2 hydrogen atoms bind with NAD, right? PGAL is oxidized and with inorganic phosphate to get converted into BPGA. Phosphoglycerate converted into this phosphoglyceric acid. The conversion of BPGA to 3-phosphoglyceric acid, 3PGA, 1 ATP is formed. Is also an energy yielding process because we get 1 ATP. This energy is trapped by formation of ATP. Another ATP is synthesized during the conversion of PEP, means phosphoenol pyruvate, to pyruvic acid, right? Can you then calculate how many ATP molecules are directly synthesized in this pathway? Directly synthesized 4 ATP, right? From one glucose molecules, 4 ATP are directly synthesized. Synthesis means formation. So 4 ATPs are formed, but 2 is used, so we gain only 2 ATP. Pyruvic acid is then the key product of glycolysis. What are the product? Product means what? Pyruvic acid, which is already 3 carbon molecule. What is the metabolic fate of pyruvate? Now second question. What is the metabolic fate of pyruvate? It is depend upon the cellular need. This depend on the cellular need. There are 3 major ways in which different cells handle pyruvic acid produced by glycolysis. These are lactic acid fermentation. First is given as for example in our muscles. When lactic acid is formed, no CO2 is released, right? And lactate dehydrogenase enzyme is used. Lactic acid formed, lactate dehydrogenase enzyme is used. Alcoholic fermentation like yeast, it forms ethyl alcohol, ethanol, and 2 CO2 is released. In ethanol, CO2 is released. And aerobic respiration, or this pyruvic acid when it goes into the mitochondria, so aerobic respiration, Krebs cycle and link reaction is there, right? So we have three parts. First one is lactic acid formation, second one is alcoholic fermentation or ethanol fermentation. Alcohol, ethyl alcohol, we also said them ethanols, right? And the third one is aerobic respiration. When this pyruvic acid enters into the mitochondria, now aerobic respiration is. Fermentation takes place under anaerobic condition in many prokaryotes and unicellular eukaryotes. Fermentation takes place, right? And as for example, for prokaryotes, any prokaryotes in which fermentation takes place? Anaerobic bacteria, they are the example of this fermentation, right? Second, eubacteria and unicellular eukaryotes, unicellular eukaryotes. An example is there, like yeast. Yeast is a unicellular cell, Saccharomyces cerevisiae. For the complete oxidation of glucose, CO2 and H2O, complete oxidation, what is the product? We have CO2 and H2O. However, organisms adopt Krebs cycle, which is also called as aerobic respiration. This requires O2 supply. O2, what is the importance of O2? O2 very little, very limited function is there. It is only a hydrogen acceptor, but it is important, very important. Without oxygen, it is not completed, right now. A fermentation is given. First fermentation is given here. Fermentation means partially oxidation. In fermentation, say by yeast is given here. The incomplete oxidation of glucose is achieved under anaerobic condition by set of reactions where pyruvic acid is converted to CO2 and ethanol. Set of reactions, first 10 set of reactions are in glycolysis. Then we have 2 set of reactions. So first pyruvic acid is converted into acetaldehyde, and this acetaldehyde is converted into ethanol plus CO2. So acetaldehyde is converted either in the formation of lactic acid or in the formation of this one, right, ethanol. The enzymes pyruvic acid decarboxylase and alcohol dehydrogenase. What enzymes are used here? Because CO2 is released, CO2 is released means decarboxylation is there, and what happens? Alcohol is formed. So we said them alcohol dehydrogenase. Catalyze these reactions. Other organisms like some bacteria produce lactic acid from pyruvic acid. Bacteria, as far lactic acid is from curd, lactobacillus, they are the example, right? Produce lactic acid from pyruvic acid. The step involved are shown in figure 12.2. That is this glycolysis is converted. Same. And another one is given here. That is no CO2 release. If CO2 is not released, so decarboxylase enzyme is not present. Lactic acid is formed, so we said them lactate dehydrogenase. Alcohol is formed, we said them alcohol dehydrogenase. CO2 is released, we said them decarboxylase enzyme. And CO2 is used, carboxylase enzyme, right? So DE used for releasing. In animal cell, okay, during exercise, when oxygen is inadequate, insufficient for cellular respiration, pyruvic acid is reduced to lactic acid by lactate dehydrogenase. The reducing agent is NADH plus, which is reoxidized to NAD in both process. Look here. Now, when we consider this one, reactions, right? What happens here? This one is our pyruvic acid is formed, glycolysis, right? Glucose, glyceraldehyde 3-phosphate. Now 3-phosphoglycic acid formed, PGA is formed. Then PEP is formed. Then pyruvic acid is formed. Now we have 2 steps here, anaerobic respiration. First, lactic acid formation, NADH, this one N, this NADH. NADH, which is formed here, na? It is used here, right? For electron transport system, they have no. They need not electron transport system, right? So this NADH is used in the formation of lactic acid, and now one new NADH formed, and this NAD is used here. Is cyclic for NAD. NAD, which is released from here, it is used as a hydrogen acceptor. Now this NAD, it is used for hydrogen donor here, right? In another case, same process is there. NAD, this NAD, it is used and NAD is released. This is also this one, right? What is the difference between these two? Pyruvic acid, formation of lactic acid, CO2 absent. No CO2 is there. That means decarboxylase enzyme is not used. Lactic acid is formed. Which enzyme is used? Lactate dehydrogenase. And in formation of ethanol, ethanol means ethyl alcohol, wine, beverage industry, right? Ethyl alcohol, CO2 is also released. CO2 is released, enzyme is decarboxylase enzyme. And alcohol is formed. We said them alcohol dehydrogenase enzyme, right? One thing is also given here. In both lactic acid and alcohol fermentation, not much energy is released. Only 2 ATP is released, right? Less than 7% of energy in glucose is released, and not all it is trapped at high energy bond of ATP. Less than 7% only energy is released, right? Given here, how many? Less than 7% energy is released. Also, this process are hazardous. Either acid or alcohol is produced. Why it is hazardous? Because either acid or alcohol is produced. When the percentage of this acid or alcohol is higher, then it is toxic for organisms. And given how many? 13%. Also process is hazardous, either acid or alcohol. What is the net ATP that is synthesized? That is how many net ATPs? We have 2 ATPs. Calculate how many ATP are synthesized and detect the number of ATP utilized during glycolysis, right? So we know very well 2 are inside, 4 are inside, and 2 are utilized. That means we have only 2 ATP gain. When one molecule of glucose is fermented to alcohol or lactic acid, okay, we know very well 4 synthesized and 2 gain. Yeast poison themselves to death when the concentration of alcohol is reaches about 13%, right? Now one question is given here. What then would be the maximum concentration of alcohol in beverages that are naturally fermented? What is the maximum concentration? It is 13%, right? But when you see the wine, right? So that means either rum, not wine. Wine and beer, it has less. It has less than 13, 8% alcohol is there. But in rum, whiskey, brandy, it has 25 to 40% alcohol is there, right? So how 25 and 40% of alcohol is formed? Because when it is near about to 13, yeast becomes dead. So they cannot form more than this concentration, right? How? Through distillation. And distillation means water is evaporated, only we have alcohol, right? So distillation. So what then would be the maximum concentration? It is 13% maximum, right? Near about to 13%. How do you think alcoholic beverage of alcohol content greater than this concentration are obtained? Through distillation, right? Answer is through distillation. What then is the process by which organisms can carry out complete oxidation of glucose and extract the energy stored in synthesize a large number of ATP molecules needed for cellular metabolism, and this one is called aerobic respiration. In eukaryotes, these steps take place within the mitochondria and it requires oxygen. So it is called aerobic respiration, and aerobic respiration is the process that leads to a complete oxidation of organic substances, either glucose or protein or fat or other organic acids, right? In the presence of oxygen and release CO2, water and energy. large amount of energy present in the substrate. Okay, this is very common in higher organisms. Now here next, aerobic respiration. For aerobic respiration to take place within the mitochondria, the final product of glycolysis, pyruvate, this is pyruvic acid, we also said them pyruvate. Pyruvate is transported from cytoplasm into the mitochondria. Now mitochondria is double membrane structure, right? So double membrane structure means, look here, this one is mitochondria and it is double membrane structure. This one is outer lamellary, this one is inner lamellary, right? In between we have a space and this one is called intermembrane space. It is called intermembrane space, right? Sometimes we said them also perimitochondrial space. That means peripheral, right? There is a difference in between these two membranes. They are not same because inner mitochondrial membrane, they have more proteins and this, it has more cholesterol, right? More proteins because they have complexes, 5 different complexes for ETS, electron transport system, right? And it has less because it is porous. It is porous. So pyruvic acid come from cytoplasm through this pores, enters into the perimitochondrial matrix or intermembrane space, and in this space link reaction occurs. Link reaction means what? Here pyruvic acid, when it enters into the intermembrane space, so this pyruvic acid, it meets with CoA, coenzyme A. It is a vitamin B, right? CoA enzyme. Second, NAD, right? NAD, and forms what? Forms acetyl CoA enzyme. Second, one CO2 is released and one NADH2 is formed, right? First CO2 is released only in mitochondria. Glycolysis process, no CO2 is released, right? Okay, now question is there, fermentation. In case of alcoholic fermentation, CO2 is released. It is inside the cell or it is outside the cell? CO2 is released in alcoholic fermentation. Alcohol fermentation, CO2 is released. Where it is released? In the cytoplasm or outside the cytoplasm? Because here given first CO2 is released in mitochondria, no CO2 is released in cytoplasm. Alcohol is formed inside the cell or outside the cell? That is first tell me. Yeast cell P forms the alcohol, right? Inside the cell or outside the cell? This respiration take place inside the cell or outside the cell? Outside the cell. CO2 is formed inside cell and released outside. Ethyl alcohol is formed, released outside. Inside the Cl, no alcohol is there, no CO2 is there. This one is correct. Inner membrane, intermembrane space. Intermembrane space, which reaction is there? Linking reaction is there, in which pyruvic acid is converted into acetyl CoA enzyme, right? And this, now we have this one. One thing or another thing. All the respiratory enzymes are present in mitochondrial matrix except one. And which one is not present? It is succinic dehydrogenase. So succinic dehydrogenase, it is not present in mitochondrial matrix. It is present dehydrogenase. It is present only in inner membrane, right? Inner membrane. And this succinic dehydrogenase, it is responsible for transportation of FADH2, right? Okay, here given. For aerobic respiration to take place within the mitochondria, the final product of glycolysis, that is pyruvic acid, is transported from cytoplasm into the mitochondria. The crucial events in aerobic respiration is first, the complete oxidation of pyruvic by the stepwise removal of all the hydrogen atoms, leaving 3 molecules of carbon dioxide. Because pyruvic acid is 3 carbon molecule, only 3 carbon dioxide is released, right? The passing on the electrons removed as part of hydrogen atoms to molecular oxygen, which is simultaneous synthesis of ATP. So this one Krebs cycle and this one is ETS, electron transport system. What is the interesting to note is that the first process take place in the matrix. First process means this Krebs cycle, right? First process take place in the matrix of mitochondria, while the second process is located on the inner membrane of mitochondria, right? ETS located on the inner membrane of mitochondria, and Krebs cycle into the matrix, mitochondrial matrix. Pyruvate, which is formed by glycolytic catabolism of carbohydrate in the cytosol, that is cytoplasm, after it enters mitochondrial matrix, undergoes oxidative decarboxylation. Why we said them decarboxylation? Because CO2 is released, right? So we said them decarboxylation. Oxidative means oxygen is used, right? By a complex set of reaction catalyzed by pyruvic dehydrogenase, right? The reactions catalyzed by pyruvic dehydrogenase requires the participation of several coenzymes, including NAD+ and coenzyme A. Now here reaction. What is here given? Pyruvic acid plus coenzyme A plus NAD in the presence of one. This is Mg+, right? Na? Mg+ means what? Magnesium or manganese? Magnesium. Now if pyruvate dehydrogenase. Dehydrogenase, why it is called dehydrogenase? Because pyruvic acid release hydrogen. So we said them dehydrogenase. D means releasing. What forms? Acetyl-CoA enzyme plus CO2 plus NADH2. So another NADH is formed here. One NAD is formed in glycolysis when we consider only aerobic respiration. One NAD, two NAD in glycolysis, right? Now from one pyruvic acid, one NAD formed, two pyruvic acid, two NAD is formed here. Now 4 NADs already formed here, and this reaction is called linking reaction. This one is linking reaction. During this process, 2 molecules of NADH are produced from the metabolism of 2 molecules of pyruvic acid. So from one pyruvic acid, 1 molecule of NAD is formed. Now the produced from one glucose. One glucose means 2 pyruvic acid, 2 pyruvic acid means 2 NADs, correct? The acetyl-CoA then enter the cyclic pathway, that is Krebs cycle. Tricarboxylic acid cycle, we said them also then, name more commonly called as Krebs cycle after the scientist, this scientist, Hans Krebs, who discovered or first elucidated. Not this one, TCA cycle. Shortly we said them TCA cycle. It starts with the condensation of acetyl group, acetyl-CoA enzyme, and the first acceptor who is the OAA, oxaloacetic acid. Oxaloacetic acid, this is 4 carbon molecule or 4 carbon molecule. Now acetyl-CoA enzyme, how many carbon molecules are there? Only 2, because pyruvic acid have 3 carbon molecules and one CO2 is released. So one carbon is released in the form of CO2. Now it has only 2 carbon, right? So this 4 OAA 4 carbon molecule plus acetyl-CoA enzyme 2 carbon molecule, it becomes, when they combine, it becomes 6 carbon molecule, and this carbon molecule is citric acid, right? And water to yield citric acid is formed. The reaction is catalyzed by the enzyme citrate synthetase, because synthesis is there. Citric acid is synthesized, so it is called citrate synthetase, and a molecule of OA is released. Now it is released. Where it is released? In the mitochondrial matrix, right? It is used after some time. It is used in this Krebs cycle, right? Citrate is then isomerized to isocitrate. So first citrate, then it convert into cis-aconitic acid, and then cis-aconitic acid converted into isocitrate, right? Intermediate substance is there, cis-aconitic acid, right? And then after isocitrate is formed. Here it is only directly given citrate is assimilated into isocitrate. It is followed by two successive steps of decarboxylation. Decarboxylation means CO2 is released, right? Leading to the formation of alpha ketoglutaric acid. Alpha ketoglutaric acid, it is the intermediate molecule. So before the formation of alpha ketoglutaric acid, we release one CO2, right? So what happens here? Look, citric acid is formed, then cis-aconitic acid is formed. Citric acid is formed, then after we have cis-aconitic acid is formed, then it converted into isocitric acid is formed. Isocitric acid is converted into oxalosuccinic acid. Oxalosuccinic acid, or we said them oxalosuccinate also, like citric acid, citrate, oxalosuccinic acid. And the formation of oxalosuccinic acid, we have 1NAD. It is converted to NADH2, H plus. And now this oxalosuccinic acid is converted to alpha ketoglutaric TRIC acid or glutarate, and here one CO2 is released. One CO2 is released, right? What is given here? Look, it is followed by two successive steps of decarboxylation. CO2 is released, leading to the formation of alpha ketoglutaric acid, right? And then alpha ketoglutaric acid is then converted into which one? Succinyl CoA enzyme. And in this process, another CO2 is released. Not only CO2 is released, but NADH2 is also formed. Right? So look here, alpha ketoglutaric intermediate substance. Why it is called intermediate? Because just before one CO2 is released, just after one CO2 is released. Now how many CO2s are released from pyruvic acids? One in linking reaction, right? And two here. All the CO2 is released. Only 3 carbon, right? Now here look here. This one is 6 carbon molecule. Now alpha ketoglutaric 5 carbon molecule. Now this become 4 carbon molecule, right? Now this succinyl CoA enzyme, it is converted into succinic acid. We will read this one, है ना? Succinic acid, it is also 4 carbon molecule, and one GTP is formed, right? Then succinic acid converted into which one? Malic acid. Malic converted to fumaric acid, and one FAD is formed, right? Lastly, oxaloacetic acid is formed. When oxaloacetic acid is formed, one NAD is also formed, right? So how many NAD are formed? Look here, one NAD is formed here, another one here, right? And one in the formation of oxaloacetic acid, OAA. So 3 NAD is formed. 3 means from 1 pyruvic acid. So 2 pyruvic acid, 6, right? And 6 are formed here on 1, 2, 3. Krebs cycle. Link reaction 2. And glycolysis 2. Right? How many NAD are formed? 10 NAD are formed. And 10 NAD means how many ATP are formed? Because single NAD 3 ATP. So 10 NAD H2 30 ATP. Right? One FAD is formed. That means from 2 FAD is formed from 2 pyruvic acid. So 2 FAD means 4. How many ATP are formed? ATP in glycolysis 2 ATP we gain. Here GTP is formed. Where GTP? This GTP. 2 GTP. GTP means equivalent to ATP. Right? So how? 30 from NADP, NADH2. Then 4 from FADH2, ATP2, GTP2. it becomes 34 plus 2 plus 2, 38 ATP. Understand na? Okay, after this one, after Krebs cycle, I explain you the blank page. Okay. Then succinyl coenzyme is in the remaining step of citric acid cycle. Succinyl co is oxidized to OAA, oxaloacetic acid. The cycle to continue. During this conversion of succinyl coenzyme to succinic acid, a molecule of GTP is synthesized. Conversion of succinyl coenzyme to succinic acid, one GTP is formed, right? And this is a substrate level phosphorylation. In a coupled reaction, GTP is converted to GDP with the simultaneous synthesis of ATP and ADP, right? Also, there are 3 points in the cycle where NADPH is formed and 1 point where FAD is reduced to FADH2. The continued oxidation of acetyl coenzyme via TCA cycle requires the continued replenishment of oxaloacetic acid. All oxaloacetic is formed. The first member of the cycle, oxaloacetic acid, the first member of the cycle, right? In addition, it requires regeneration of NAD, FAD from NAD and FADH2 respectively, and the summary equation for this is given here, right? And equation is given here. Cycle is given here. Look, pyruvic acid plus 4 NAD plus FAD plus 2H2O plus ATP. How many? In mitochondrial matrix, 3 CO2 is released, right? NADH, 4 NADH is formed. One in linking reaction, 3 in Krebs cycle, right? 4 H plus ion is released. FAD H2 is formed. How many? 2 FAD is formed, right? Now into and ATP is formed, right? So from 1 pyruvic acid, 3 CO2, 4 NAD, 4 H plus ion, 4 FAD and ATP is formed. Citric acid cycle is given here. Right, okay. We complete this. We have till now seen the glucose has been broken down to release CO2 and 8 molecules of NADH. 2 FADH has been synthesized beside just 2 molecules of ATP in TCA cycle. You may be wondering why we have been discussing respiration at all. Neither O2 has come into the picture nor promised large, because in this whole process oxygen is not involved anywhere else, right? Oxygen is not involved, but we need oxygen for respiration. What is the importance of oxygen? In electron transport system, we use oxygen in electron transportation, because our oxygen is the ultimate acceptor of hydrogens, electrons, right? You may wondering, okay, neither O2 has yet. Also, what is the role of NADH and FADH2 that is synthesized? Let us now understand by the ETS system. Here, electron transport system. So first here, very simply I explain you the Krebs cycle, right? First glycolysis is there, na? Can you tell me the first? We have glucose, right? Now glucose is converted into glucose 6-phosphate. Very good. Glucose 6-phosphate, right? Which enzyme is used? Hexokinase. Hexokinase enzyme is used. ATP is used. Phosphate. This phosphate come from ATP. That means we use ATP, and this ATP is converted into ADP. One phosphate come into this glucose molecule. Now glucose 6-phosphate is converted into fructose 6-phosphate. Fructose 6-phosphate. Fructose 6-phosphate. Which enzyme is used? Isomerase, because it is an isomer. So isomerase enzyme. ASE, isomerase enzyme. Right? Now fructose 6-phosphate is converted into fructose. Fructose 1 ATP is also used here. So fructose 1,6 bisphosphate, right? 1,6 bisphosphate. Now fructose 1,6 phosphate is further converted into, it is splitted, right? It is splitted in two parts. One is our PGAL, shortly we said na, phosphoglyceraldehyde, and another we have DHAP. DHAP means what? Dihydroxyacetone phosphate. Look here, this is 6 carbon molecule. This one is also 6 carbon molecule. This is also 6 carbon molecule. 6 carbon molecule. But this one is 3 carbon molecule, and this is also 3 carbon molecule. Triose sugar. These are triose sugar. So we said them also triose phosphate, because phosphate is there. Triose phosphate. Phosphoglyceraldehyde. That is called dihydroxyacetone phosphate. Right? Now what enzyme here is used? Aldolase enzyme. Right? Now here this PGAL, I write from there, right? This PGAL. This PGAL. Now this PGAL, it is called 3-PGAL, 3-phosphoglyceraldehyde, right? 3-PGAL or 3-dihydroxyacetone phosphate. Now this dihydroxyacetone phosphate is further converted into PGAL, right? For further reactions. Now PGAL is converted into BPGA, bisphosphoglyceric acid or bisphosphoglycerate, right? But here what happens? By using of one inorganic phosphate, that is H3PO4, inorganic phosphate, right, and H2O, it forms NAD plus, is converted into NADH plus H, NADH2, right? What is the full form of NADH? Nicotinamide adenine dinucleotide, right? P is not there, so I cannot write phosphate. NADP means phosphate. Okay, now PGA is converted into 3-PGA, and this BPAG means 1,3-BPGA, bisphosphate, right? Now look here, one phosphate is released. Where this phosphate goes? This is used in the formation of ATP. ATP is formed, right? Here one ATP is used in this process. One ATP is used. Now it becomes 1,6 bisphosphate. Now this PGA is converted into PEP, phosphoenol pyruvate enolase enzyme. First one thing is there, I am directly right here, 3-PGA is converted into 2-PGA, right now. Otherwise our steps will one less step is there. We have to complete the 10 steps. So 3-PGA is converted into 2-PGA, and enzyme which is used here, it is mutase. Mutation is there, so it is called mutase enzyme, right? And now 2-PGA is converted into PEP, phosphoenol pyruvate, right? And enzyme is used here, enolase, ASE. And now PEP is converted into pyruvic acid, right? This is 2 multiply means 2 molecules of 3-PGA, 2 molecules of 2-PGA, 2 molecules of 2-PEP, and 2 pyruvic acid is formed. And this pyruvic acid is also 3 carbon molecule. This is 3 carbon. This one is also 3 carbon. 3 carbon. 3 carbon. All are these 3 carbons. How many steps are there? Now which are the irreversible? All these are reversible, but this one, glucose, this one is irreversible. This one is reversible, but this one is irreversible, right? This one is reversible, we can draw this one, so it can convert, no? This is reversible. And then after we have 10th step, this one, This one is also irreversible. All these are reversible. Right? First 1, 3 and 10 reversible steps. 1, 3 and 10. Right? Now this pyruvic acid, it is undergoing in the fermentation, that means alcoholic formation of alcohol or formation of lactic acid. In fermentation, what happens? In fermentation, now we have pyruvic acid. This pyruvic acid, it is converted into acetaldehyde. And this acetaldehyde is converted into either lactic acid or second is our pyruvic acid. Same process, acetaldehyde, but here what forms? Ethyl alcohol, C2H5OH and CO2. Two molecules, 2 C2H5OH and 2 CO2 from two pyruvic acid. Right? So here which enzyme is used? Because carbon dioxide is released, it is called decarboxylase enzyme. Here alcohol is formed, ethanol is formed, it is called alcohol dehydrogenase. Lactic acid is formed, which enzyme is used? Lactate, LAC, yes, lactate, PAT, and dehydrogenase. Right? Another what is used here? We use here NADH, NADH2. It is converted into NAD in both process, NAD plus. Here also NADH2 is used, and what is released? NAD plus is released. Right? And this NADs come from glycolysis. Right? Na? NAD. As for example in yeast, and it is example of our muscles. Which muscles? Skeletal muscles only. Right? In voluntary and cardiac muscles, they have no this. The lactic acids are not produced. Only skeletal muscles they produce lactic acids. And now we have this here pyruvic acid, which is in cytoplasm, right? Now from cytoplasm it comes into intermembrane space, and in intermembrane space this pyruvic acid it combined with CoAA enzyme. Actually this one is vitamin B, right? Plus NAD, right? Now what happens here? We obtained acetyl CoA enzyme plus NAD. This one is D, right? H plus H and CO2. It is gas, so it is released plus, right now? Plus CO2. Look here, this pyruvic acid is 3 carbon molecule, right? And acetyl CoA enzyme is 2 carbon molecule. Why? Because 1 CO2 is released, 1 carbon is released here. Right now, 1 carbon is released here. So this one is called linking reaction. This one is called link reaction. link reaction. Why it is called link reaction? Because pyruvic acid is not used in Krebs cycle directly. First, it has to convert into acetyl-CoA enzyme. First, it has to release one carbon molecule. Because oxaloacetic acid only binds with acetyl coenzyme, which is 2 carbon molecule, right? Now, after this link reaction, this acetyl-CoA enzyme, it is captured by the protein, extrinsic protein, and this extrinsic protein, it post or transport acetyl-CoA enzyme into the matrix, and matrix all the respiratory enzymes are present except one. Which one? Succinic dehydrogenase, right? And this enzyme is present only in inner membrane of mitochondria. So Krebs cycle, in Krebs cycle we have oxaloacetic acid, it is the receptor, right now, and this one is 4 carbon molecule. Now this oxaloacetic acid, it binds with acetyl-CoA enzyme, which is come from link reaction, right now, and this one is 2 carbon molecule. Now they forms citric acid. The first molecule which is formed here, it is citric acid. So it is also called as citric acid cycle, right now. First molecule which is formed in photosynthesis, it is 3 carbon molecule. So we set up C3 cycle. This is PGA. RUBP plus CO2 H2O, it becomes PGA, phosphoglycic acid, right now. First product which is formed in C4 cycle, it is oxaloacetic acid. So we set them C4 cycle. So first product name is given according to first one. It is citric acid. So we set them also citric acid cycle, right? Now this citric acid cycle, it becomes 6 carbon because 2 plus 6, 4 equal to 6, right? And this citric acid cycle convert into cis-aconitic acid, right? And this cis-aconitic acid, it is converted into isocitric acid. All these are 6 carbon molecules. Now isocitric acid is converted into oxalosuccinic acid, and in the formation of oxalosuccinic acid we have NAD plus NADH2. I write here H2, H plus H. You should write H plus H right now. Is formed. First product. Then oxalosuccinic acid is converted into alpha ketoglutaric acid. Alpha ketoglutaric acid. In the formation of alpha ketoglutaric acid, one CO2 is released. First, in Krebs cycle, it is the first carbon which is released. Another carbon is released in linking reaction, right? Now, alpha ketoglutaric acid is converted into succinic CoA enzyme, right? This, in the formation of this one, this CoA, which is released here, where this CoA is released, right now, here CoA is released, and this CoA is binds with succinic, this one, right? And succinic CoA is formed. One CoA is formed, one CO2 is released here. This CO2 is one CO2 released, and also one NAD plus NADP is formed. NADH2 is formed. CoA enzyme attached, CO2 release, NAD is converted to NADH2. So ketoglutaric is called intermediate because on both sides CO2 is released. Now succinic CoA enzyme, it is converted into succinic acid or succinate. Succinic acid or succinate. And in the formation of succinic acid, one GTP is formed. GDP is converted into GTP. And this GTP, it is equivalent to ATP. Further, it is used in the formation of ATP, right? Succinic acid is converted into malic acid. First, fumaric acid. Fumaric acid, right? And in the formation of fumaric acid, one FAD is formed. FAD, and it is converted into FADH2 is formed. Right? FADH2 is formed. Now, fumaric acid is converted to malic acid. Malic acid, and one H2O is released, right? And this malic acid is converted into oxaloacetic acid. And in the formation of oxaloacetic acid, another NAD plus is formed. NADH plus is formed, or NADH2 is formed, right? No? NADH2 is formed. Now look here, one CO2, this oxalosuccinic acid, it is 6 carbon molecule. Alpha ketoglutaric acid, it becomes 5 carbon because one carbon dioxide is released, right? And now this succinyl coenzyme, it becomes 4 carbon molecule because one CO2 is also released. Succinic acid, 4 carbon molecule, fumaric, 4 carbon. Now all these other are 4 carbon molecule, and alpha oxaloacetic acid is also 4 carbon molecule, right? So how many? Look, how many NAD is formed here? 1, 2, 3. So 3 NAD. Look, 3 NADH2 is formed in Krebs cycle, right? So 3 means what? 2 multiplied by 3 because 2 pyruvic acid. We have 2 pyruvic acids now. Now, how many NAD is formed in linking reaction? Link reaction, we have 1 NAD is formed. So link reaction is 2 NAD is formed. From 2 to 1 NADH2 multiplied by 2 because 2 pyruvic acid is there. Now, how many NAD is formed in glycolysis? Glycolysis, NAD, 1 NAD is formed, NADH2 is formed, multiply by 2 because 2 phosphoglycic acid is there, right? NAD is formed after phosphoglycic acid. So that means what? 2 multiplied by 3, 6. 2 plus 2, total we have 10 NADH2 is formed, right? Or I can write here 2 multiplied by 5, right? 10, right? Okay, this is the NADH2. Now FAD, we have only one FAD, this one, right? One FAD is formed. How many FAD is formed? FADH2. Directly we can write here 2 FADH2 is formed in Krebs cycle due to the 2 pyruvic acids. And how many GTP is formed here? 1, that means 2 GTP is formed. 2 GTP is formed in Krebs cycle. And how many ATP is formed? We gain in glycolysis 2 ATP we gain because it is synthesized, 4 ATP is synthesized but 2 is used. We deduct 1, 4 minus 2. This 4 minus 2, we have only 2 ATP, right? Now we have how many NAD? 10. That means how many ATP is formed? Multiply by 3, 30 ATP, right? FAD2 multiply by 2, we have 4 ATP. 2 GTP means what? Multiply by 1, 2 ATP, and 2 ATP means multiply by 1, it is 2 ATP. Now total 30 plus 4, 34, and plus 2 plus 2, it is 38 ATP is formed. So in total, glycolysis, link reaction and Krebs cycle, right? Correct. Now come to the ETS. Electron transport system, ETS, or oxidative phosphorylation. Now here it is given oxidative phosphorylation. Oxidation means by using oxygen. ATP, formation of ATP by using oxygen. The following steps in the respiratory system are to release and utilize the energy stored in NADH and FADH. This is completed when they are oxidized through the electron transport system and the electrons are passed on to O2, resulting in the formation of H2O. We have H plus ion. All these have NADH2, FADH2, they have H plus ion. And oxygen is also come from breathing or respiration. So this oxygen is used to consume this one, to accept this H plus ion and formation of H2O. Are passed on O2, resulting in the formation of H2O. Right? So this hydrogen, they are used in the formation of H2O. The metabolic pathway through which the electron passes from one carrier to another carrier, they are called as electron transport system. Right? And it is present in the inner mitochondrial membrane. That's why inner mitochondrial membrane has more proteins. These are proteins, enzymes. Electrons from NADH produced in the mitochondrial matrix during citric acid cycle are oxidized and NADH be oxidized by an NADH dehydrogenase. Why said dehydrogenase? Because hydrogen is released. Carbon dioxide is released, we said them carboxylase. Hydrogen released, dehydrogenase. Right? And this one is called complex 1. We have 5 compounds. Complex 1 is NAD dehydrogenase. We said them FMN dehydrogenase. Right? Or simply NADH dehydrogenase. Complex 2nd, this is FADH dehydrogenase. Complex 3rd, we have B and C. B and C1, not C. B and C1, cytochrome B and C1, right? And complex 4, that is cytochrome A, A3 and copper, Cu. And complex 5th, it is ATP synthetase, formation of ATP. We have 5 complexes, correct? Complex 1st is NADH dehydrogenase, right? And electron are then transferred to ubiquinone, UQ. Shortly we said them UQ, right? Now, ubiquinone located within the inner membrane. They are electron transporter. Ubiquinone also receives reducing equivalent via FADH2, right? Complex 2nd, which is called complex 2nd. That is generated during oxidation of succinate in the citric acid cycle. They are generated. Which are generated? FADH are generated in the formation of succinic acid. FAD is formed, right now, when fumaric acid. The reduced ubiquinone or ubiquinol is then oxidized within the transfer of electrons to cytochrome C via cytochrome Bc. Look here, cytochrome C, this one is mobile. That means it is movement. It has movement. So it took electron from which one? Complex 3 and transport this electron to complex 4. Which one? This cytochrome C. Cytochrome C, it is for mainly transportation of electron. And this cytochrome C, it is present outside the inner membrane. Look here, now here, this one is our inner membrane. This figure, if you understand this figure, you will not read this one whole. What is given here? This one is our inner membrane, lipid bilayer, right? And these are the proteins, right? Now look, intermembrane space. So this one is outside the center, right now, in between 2, and this one is matrix, central region. H plus ion, concentration of H plus ion is more where? Toward inner or toward outer? Toward inner or outer. Concentration of H plus ion is more in outer, because look, all the arrow comes outside the, right? So concentration of H plus ion is more in inner membrane, that is outside the membrane. But in case of grana, what we read there? In lumen, concentration of H plus ion is more. That is the opposite direction, right? Look here, this one is our first complex. Complex first, NADH dehydrogenase. So here NADH, which is come from our glycolysis or link reaction or Krebs cycle, and H plus dehydrogenase enzyme is there. So these H ions, they are removed from this NAD and comes this FMN, flavin mononucleotide, right? Not dinucleotide. it capture this H plus ion and transport this H plus ion outside this. This one is act as hydrogen pump, right now, but it's not a hydrogen pump. Act as hydrogen pump is this one. Which one? ATP synthetase. Look here, H plus ion from matrix transported to intermembrane space. H plus ion of NADH through first complex, right? And this through this one, 2 electron is also come with this H plus ion. H plus is proton, right? Electron. Now this electron is transported. This one is FeS, ferrous sulfate, right? And this transport this electron to ubiquinone. Now these electrons are transported to the this one. This one is complex third. This one is complex third, not second. Second is after this, right? So this electron comes to the complex third. Okay. Now see this one second. So what is the function of this complex? Very easy thing is there. Only the function of this complex is to transport the hydrogen which come from NADH, right, to the from inner to outer, because Krebs cycle occurs in the matrix, right? And after the Krebs cycle, NADH released into the matrix, and this NADH, this hydrogen, it is transported from inner matrix to peripheral matrix, perimitochondrial matrix, right? Now look here, complex second. This one is succinate fumarate, succinate dehydrogenase. It is present only in mitochondria membrane, inner mitochondria, not in the matrix, right? And what one thing is there, it is unable to transport the hydrogens which come from FADH2 outside the membrane. It transport this H2, UQH2 to this complex third. Electron plus hydrogen both are transported to ubiquinone to complex third. And this complex third transfer this hydrogen to outside. Look this outside. There is no hydrogen come outside, right? So this has this first complex first, it has the capacity, it can transport across the membrane, but complex second, it has no capacity to transport across the membrane, transport the hydrogen ion across the membrane. So it transport hydrogen ion to ubiquinone, and this ubiquinone transport this hydrogen plus electron to the complex third. Complex third transport this hydrogen outside the membrane, right? So 4 hydrogen molecules come here, 4 come here. How many? 2 hydrogen molecules are. How many? 8 molecules are transported. Now look here, cytochrome 6C. It is where it is located, outside the or inside the membrane? Outside the membrane, right? It is located in inner membrane space, outside the inner membrane, and it is responsible for the transportation of electron, not hydrogen, electron. Now this electron, this electron, it is transported to complex 4. These electrons which come from hydrogen, they are transported to complex 4. Complex 4 means what? Cytochrome A, cytochrome A3 and copper. Cytochrome A, cytochrome A3 and copper is there. Sum is copper, right? And look here, this side H2 plus O2, it forms H2O plus 2 H plus ion are also released. So this is also responsible for the transportation of extra hydrogen ion, which is not that is converted into water toward the outside. H2O is there. How many H plus ion here? 10, right? And now look here, all these H plus ions, they are, because here concentration is more, only one pump is there. This one is pump F0, F1, and the same pump is also present in lumen, right? Thylakoids. So all the hydrogen which comes this side, they pump out, right, toward the inner side. So when they are pumping, so 4 hydrogen atom, when pumped from F0, they form 1 ATP, right? And ATP is formed, and H comes to this side. This H binds with oxygen and forms H2, water. So what is the function of O2? O2 is very limited function. It acts as final acceptor of H plus ion, right? Why it is vital? Because if O2 is absent, That means hydrogen is not bound with any one molecule. So this process is not completed, right? Suppose that I give you some money, right, and said give it to the office person. So that means office person is important to send this money. This process is completed when the office person is there. No office person means what? This is lost, right? So oxygen is the ultimate acceptor of oxygen, and this is given here. Electron to cytochrome C via cytochrome BC1. BC1 means complex third, right? What are the VR complex? First complex means what? NADH dehydrogenase, right? Sometimes I said FMN NADH dehydrogenase, right? Second, succinic dehydrogenase. Succinic dehydrogenase, this one, right? Third one is our cytochrome BC1 complex. So cytochrome B, cytochrome C complex is there. Cytochrome BC1 complex. This one third. Fourth one is cytochrome A3 copper, right? And cytochrome fifth ATP synthetase, right? Here, what is cytochrome 6? Cytochrome C. What is the function of cytochrome C? Cytochrome C is present outside the membrane. It acts as mobile or transporter of electrons, right? All the hydrogen ion which comes out from this matrix, they are transported. Only one pump is there, that is F0, F1 particles. When the electron, okay, we will continue this one. UV quinone receives, okay, second. It is given also here. Cytochrome C via cytochrome BC1. Cytochrome BC1 is called complex third, right? Cytochrome C is a small protein attached to the outer surface of the inner membrane and acts as a mobile carrier of transfer of electron between complex 3 and complex 4. Between complex 3 and complex 4, this one, cytochrome C, right? Now, complex 4 refers to cytochrome C oxidase complex, which contains cytochrome A, cytochrome A3 and 2 copper centers. Right? When the electron passes from one carrier to another via complex 1 to 4, in the electron transport chain, they are coupled to ATP synthetase enzyme. This is called complex fifth, right? So who coupled with ATP synthetase? This electron, right? For the production of ATP from ADP, for the production of ATP and inorganic phosphate. The number of ATP molecules synthesized depend on the nature of the electron donor. Nature means what? Oxidation of one molecule of NAD gives rise to 3 ATP. Then after FADH2 produce 2 molecules of ATP, and GTP, we know GTP is equivalent to ATP, right? The role of oxygen is limited in the terminal stage, means final stage of the process, yet the presence of oxygen is vital. Why? Because since it drives the whole process by removing hydrogen from the system, right? So oxygen acts as final hydrogen acceptor. Unlike phosphorylation, where it is light energy that is utilized for production of proton gradient required for phosphorylation, in respiration, it is the energy of oxidation reduction utilized for the same process, and it is for this reaction that the process is called oxidative phosphorylation, assertion region. Why it is called oxidative phosphorylation? Because it requires oxygen, right? You have already studied about the mechanism of membrane linked ATP synthesis, experiment chemo-osmotic hypothesis in the earlier chapter. Earlier chapter means what? Photosynthesis. Right now, we are, as mentioned earlier, the energy released during the electron transport system is utilized in the synthesis of ATP. In old NCERT it is given a 2H plus ion, but in new NCERT it is given 4H plus ions, right? And this figure is given here. This one is F0 particle, this one is F1 particle. F1 is responsible for the formation of ATP, right? So passive transportation for H plus ion is F0 and active transportation F1 0, right? Okay, you read this one. Now respiratory balance sheet. Balance sheet means how many ATPs are formed, right? We already read this one. There is a sequential orderly pathway functioning with one substrate forming the next with glycolysis, TCA and ETS pathway following one after another. Continue alternate forms. The NAD synthesized in glycolysis is transferred into the mitochondria and undergoes oxidative phosphorylation, that is in ETS. None of the intermediate in the pathway are utilized to synthesize any other compound. All the compounds are used only in the formation of ATP, right? Only glucose is being respired here. No other alternative substrates are entering in the pathway at any of the intermediate stages in this process. Now here look, but this kind of assumptions are not really valid in a living system. All pathway works simultaneously and do not take place one after another, right? Substrate into the pathway and are withdrawn from it as and when necessary. ATP is utilized as and when needed. This is that means our metabolic activities, they are simultaneously occurs in our body. So not only one reaction is there, thousands of reactions are occurs at the same time, right? In which one reaction is this one is one reaction. And ATP is stored when we need. ATP is used. So respiration means only the formation of ATP, right? Only the formation of ATP. Hence here there can be a net gain of 38 ATP as we already read molecules during aerobic respiration of one molecule of glucose. Now it is completed now. Let us now compare the fermentation. You should read this one, right? Fermentation and this one. In fermentation there is net gain of only 2 molecules of ATP. We know very well glycolysis pays off. Okay, NAD is oxidized to NAD rather slowly in fermentation. Why slowly? Because NAD is used in the fermentation also. Amphibolic. Why it is called amphibolic pathway? Because ATP is also formed. ATP converted to ATP. ATP is formed and that one is anabolic reaction. And glucose is breakdown. That is catabolic reaction. So both reactions are formed. That means it is, yes, this one is amphibolic pathway. You know, breaking down of process within the living organisms is catabolism and synthesis is anabolism. And because in respiratory pathway it involves the both anabolism. Anabolism means what? And catabolism. Anabolism means what? Formation of ATP, right? Like one. And anabolism or catabolism means what? Breakdown of glucose molecules. It would hence better consider the respiratory pathway as an amphibolic pathway rather than a catabolic one. Commonly, initially we read respiration is a catabolic pathway because glucose molecules are breakdown. But some molecules are also formed here. All this it is given here where molecules are formed, where molecules are broken, right? So all these metabolic, catabolic, anabolic pathways are formed in like such. We have pyruvic acid. Pyruvic acid is breakdown. First glucose is breakdown. Glucose is breakdown into PGAL and that is one DHAP. That one this reaction is catabolic reaction, right? Okay, pyruvic acid breakdown in acetyl coenzyme. This one is also catabolic reaction, right? Now OAA plus acetyl coenzyme, they join with each other, form citric acid. This one is anabolic reaction, right? So we have many reactions. NAD plus NADH2 is formed. This one is anabolic reaction, right now. So we have both reactions are present in this pathway. So this one is amphibolic pathway. Also this one is given. We have 3 components: fat, carbohydrate, proteins. So this which we read, it is carbohydrate pathway, right now. Now proteins, only look, proteins monomers are amino acids. Fats monomers are fatty acids or glycerol, right? Carbohydrate monomers are simple sugar glucose. So this column we have monomers, all the monomers are there. Now this fat or fatty acid and glycerol, it has 2 doors to enter into this glycolysis cycle, right? So first in the form of dihydroxyacetone phosphate, First, it converts into dihydroxyacetone phosphate, then it directly enters into the glycolysis. And second, it is converted into acetyl-CoA enzyme. That means in the mitochondria. In cytoplasm, in the form of DHAP, another in mitochondria in the form of acetyl-CoA enzyme. Right? And in case of protein, amino acid is there. So it has also one pathway is given here in the form of pyruvic acid. It comes, right? And sometime what happens? This amino acid also forms the alpha ketoglutarate acid, right? It also directly comes to the alpha ketoglutarate acid. But here, why it is not considered here? Because alpha ketoglutarate acid, after alpha ketoglutarate acid, we have no that NAD is formed, but less amount of one NAD is formed, one FAD is formed, and one CO2 is released, right? One GTP is formed. So amino acid, two doors are there: pyruvic acid in cytoplasm, alpha ketoglutarate acid in Krebs cycle, mitochondria. Fats, fatty acids and glycerol, it has also two in cytoplasm in the form of DHAP, dihydroxyacetone phosphate, and in Krebs cycle in the form of acetyl-CoA enzyme. Respiratory question, that means CO2 upon O2, right? Simply it is given here. Depend upon the type of respiratory substrate. Carbohydrate means only one, because O2, 6 O2 is used and 6 CO2 is released. So 6 CO2 upon 6 O2 is 1. So carbohydrate is 1, right? But protein, look at this given na. This is 1 carbohydrate form. When fat are used in respiration, RQ is less than 1. Less than 1. Look at C51H98O6. This one is a fat acid, right? Fat. Plus 145 O2 is used. How many CO2 is obtained? 102 only. Less carbon dioxide release. That 102 upon 145 is less than, right now, 0.7, less than 1. When protein or respiratory substrates are raised, the ratio would be also near about to 1, but not 1, less than 1. It is 0.9, right? 0.9. Pure protein and pure fat, they are never used as respiratory substrate. This last point is very important. It should be asked, right? So pure protein and pure fat, they are never used as respiratory substrate, right?